Hello, everyone, and welcome to the ROSA Worldwide Webinar Series. My name is Mike Reed. I'm the Chief Scientist at ROSA, and I'm delighted to meet with you today. Our speaker today is Emily Nieves. She is the co-founder and CEO of Delineate, a seed stage company that collaborates with pharmaceutical organizations to leverage large language models and artificial intelligence technologies to advance model-informed drug development. Emily holds a Bachelor of Science in Biomedical Engineering or Biological Engineering from the University of Georgia, where she worked in Dr. Melissa Hallow's Quantitative Systems Pharmacology lab. Her professional experience includes the development of QSP and machine learning models across diverse therapeutic areas, including the cardiorenal system, metabolism, and cell-based therapies, with collaborations at Pfizer and AstraZeneca. During the talk today, you can ask questions by clicking on the Q&A button at the bottom of your screen. And we look forward to today's presentation. So take it away, Emily.
Great. Thank you so much for that introduction. I'm really excited to be presenting today. So just a little bit more background on Delineate. So as was mentioned, we're a multidisciplinary team of 20 plus engineers and researchers focused on applying AI to accelerating modeling workflows. And we are supported by a number of organizations, including MIT. We have some funding from NIH and a number of our investors as well. By way of a little bit of background, although I'm sure a lot of people on the call are familiar with model-based meta-analysis, I wanted to kind of set the stage on why this topic is relevant for us. So MBMA is a quantitative framework that integrates data from multiple clinical trials using mathematical models to characterize dose or exposure response relationships. So oftentimes you are collecting data from external clinical trials via publications, and then integrating that with your own internal data to help support certain decisions such as dose selection, competitive benchmarking, supporting go, no-go decisions. And here, really, the creation of those harmonized data sets is the critical bottleneck. This is the part of the process that can take months to complete. The actual model building is quite routine and pretty easy to for a pharmacometrician to conduct that. So the question here really is, can we use GenAI to assist in this stage without compromising accuracy? So that's a theme that I'll be going through a lot in the presentation, is how do we set up appropriate guardrails through design of software that allows for a scientist to be able to audit what the LLMs and the AI models are outputting so that we can ensure accuracy without sacrificing efficiency, since that is our whole goal. So our process here at Delineate is we kind of break down the individual steps of constructing a database and integrate AI at each step. And then, as I mentioned, with a specific focus on auditability and transparency. So these databases are fit for purpose. So they're not made ahead of time. They are made for the specific analysis. So that's another way that we kind of operate a little bit differently. And that allows us to not only support things like MBMA, but we've helped companies build data sets for training specialized AI models, building parameter databases for building large mechanistic models, etc. So, and you'll see that theme throughout the presentation as well, this idea of it being very fit for purpose and very flexible. So I'll walk through each of these steps, but first is conducting the systematic literature review, where you're finding the studies that have the data that's relevant in the population that you're interested in studying. And then we have a lot of tooling around extracting the data from publications, so from across the graphs and the tables and the text. And then at the end, pulling all of that data together into a data set that's ready for your analysis and tailored to the specific software that you are going to be working with. So in the first piece, we have our literature review step. So here we use AI agents to automate certain parts of this process. So the first step is the creation of criteria. So we can turn a user query into a set of criteria that then each publication can be assessed against. So in the context of MBMA, this could look like aligning to a PICOS framework where we're setting up what population we're interested in, what intervention, etc. And so here in the screenshot, you can see there's a number of criteria that each of the abstracts get assessed against. And then there is the reasoning given for why each of the criteria was met or why it was not met. And here there is some nuance, right? So we're at this part of the process only looking at the abstract. So there's room for, um, interpretation, or there's things that may be in the full text that are not available in the abstract. So here there's three options. Either the criteria is met, it's not met, or there's also a maybe option. So um, there may be that data that you're looking for in the supplement, for example, and that would get flagged for the next step of the process, and that's where the scientist would kind of take over there. So that is the first step, is finding abstracts that meet some of this criteria, and then you can filter out based on that. And then these get moved into what we call a collection. So this is where scientists are working together to kind of build up this database and really fulfill the requirements of a systematic literature review, a very rigorous review. So here, if the paper is open access, it's able to be downloaded. That kind of happens automatically. And then the LLMs will go ahead and assess the full text and any supplementary information to, again, assess against that criteria. So make sure that those inclusion criteria are met. If the full text is not available, the user can then upload the paper, go through their institution. If they have access via that publisher, can upload the full text, and then the criteria can be assessed there. And this system, again, is nice in that it's collaborative. So multiple scientists can assess a paper, make sure they agree with the AI reasoning, and then approve it for inclusion in the database or not. So again, this is kind of a hybrid AI and scientist workflow where we are trying to minimize the search space. So we don't want scientists having to read hundreds of papers, but we do want the ones that are likely to fit inclusion criteria. We do want the scientists to kind of be able to review those as a final step. So we've done some benchmarking on this search method. So we replicated the literature review portion of 3 published MBMA studies. On average, accounting for year of publication and the inclusion-exclusion criteria, we were able to find 15% more studies than manual search. But most importantly, as I mentioned, we reduced the search space by an order of magnitude. So only the papers that met or had a high likelihood of meeting certain criteria were needed to be reassessed by the scientist. So another key feature of our software is the automated plot digitization. So a lot of the data that is necessary to build these meta-analysis is behind these graphs and charts. So we have custom trained computer vision models that have been trained on a vast amount of manually digitized graphs that allow us to extract the XY coordinates from graphs such as spider plots, Kaplan-Meier curves, line plots, and waterfall charts. And we can also get the error bars and detect whether those error bars are a standard deviation, is that a interquartile range, etc. And so here our computer vision models outperform state-of-the-art models, 87% accuracy across 60 publications of different plot types. And here is another way that we are taking kind of a hybrid approach where we use language models for certain aspects, but we also use more traditional computer vision to improve accuracy. And then again, thinking about auditability and transparency, We design the system such that the scientist remains in the loop. So on the right, you can see a screenshot of what our plot digitizer looks like, and it looks very similar to web plot digitizer, the more manual method. And this is on purpose so that the scientist can look at, the data that was digitized and very easily visually see if a point was missed, if it needs to be adjusted, and those adjustments can be made really easily. And this is in contrast to if we dropped in a PDF or an image of a graph into something like ChatGPT. It may give us X, Y points in a list, and some of those may be correct, but it would be very laborious for us to go through and figure out which ones are in error and which ones are correct. So here we, again, with the design of the user interface, we try to make it really easy for the scientist to see where the AI is going wrong. And then, special to model-based meta-analysis is extracting dosing events and covariates and being able to intelligently link those to that observation data that we do get out of the graphs. So here we have multi-agent systems that extract the covariate information from across the full text and the tables. Importantly, this is regardless of specific wording used. So if a covariate is referred to in different ways across the different publications in your database, it will still pull those together. And then we link those covariates to the observation and dosing information. And then the dosing schemes are extracted again across the text, any figures in the supplement, and converted to dosing events that are, fit for setting up a data set that's ready for non-mem and model building. So calculating things like what is the interdose interval, how many additional doses were given, and capturing dose titration and all of those nuanced things that we often are doing manually via reading the publication and kind of doing those calculations ourselves. We have a multi-step process for that. So kind of wanted to highlight again, the design of the user interface such that not only is this fit for model-based meta-analysis, it's not really hard-coded for that. So the way that we've set this up is you first define a data extraction template, as we call it. And this is where you are defining the columns and the data fields that you would like to pull out from across those, the text and the table and the figures. And this allows you to really create any custom database. So again, you can set up a database for constructing an AI model, a very specific, model you're trying to train, or a parameter database, etc. And you can encode the specific format that you would like it to kind of take. So, for example, in regimen, if you want that to be just the abbreviation and not, the spelled out any extra details of regimen, you just want the abbreviation. You can set that up in this template. You can also encode very specific formatting or conventions that are unique to you and the analysis that you're running. So things like specific IDs for separating out endpoints. Again, that can be done here in this template. So this is very powerful, and this is where a lot of the bulk of the work actually is in setting up this template. It's an iterative process to kind of define the data set you're looking to create across potentially hundreds of publications. Another key challenge of building these databases is often standardization and harmonization across all of these publications, since the wording is slightly different across all of them. And at the end of the day, we need a data set that we can pull in, and each of the data fields are, if they refer to the same entity, they should be very standardized. So here we use large language models to help us standardize. So we convert things like, so as very simple example for regimen, one paper may call it once daily, and then the other papers may use the abbreviation. we can use language models to standardize all of those entries into just the abbreviation. But we also don't solely rely on large language models. Again, this is another area where we take a hybrid approach. So for things like units, we have a units library that is more manually created. And this allows us to do conversions and things like that to maintain consistent units, but we're not relying solely on large language models to kind of do those calculations, et cetera, because that's an area where you can lose some accuracy. And it's a little bit difficult for the scientist, right, to go through and audit that if they're doing multiple calculations. So that one is done a little bit more manually. Um, Similarly, we add a lot of programmatic checks of the data. So we're flagging any potentially errant data, looking at things like outliers, biological plausibility for certain, covariates or things like that. And again, this is done a little bit more traditionally, the old-fashioned way. We don't rely on large language models for that as much. So we take a more traditional approach to our guardrails. Um, and as I've been mentioning as well, all the data points and entries are quickly auditable back to the source, which allows for easy scientist oversight. And so the errors that pop up, they will tell you, the exact file, the exact line that may need to be checked again, and then it can be easily corrected within the platform, and you can just regenerate your data set in a matter of seconds there. So that was the piece on building the data set. So there's a lot there, as I mentioned, very flexible. But then once you have this data set, you can also pull it into our model building copilot. So this is within the same environment as the data extraction piece. And that's important because all the details of the publication and that data schema, that template extraction that you set up are in context. So you don't have to explain the setup of your data set to the AI before you ask it to do some exploratory data analysis, for example. It kind of understands the details of the trial and understands the formatting of your data set. And so again, here the scientist remains in the loop. It approves the plan for conducting data analysis, but then also it can help you build your final model. And so again, this is iterative. So the results are fed back in to guide the final steps. So for example, you can ask the AI agent to assist you in doing some exploratory data analysis. It will use the results. So looking at things like, the plots of a covariate versus response. And then you can look at that, and those results are fed back in to guide the model building. So it may suggest, okay, I think this covariate may be important for you to include in your model, or based on this plot, I think we should maybe consider this model structure. But of course, the scientist is there with their expertise to see if they agree with that suggestion and move forward with that. Some really exciting R&D that we've been working on lately is actually full model replication. So can we drop in a publication, a PDF of a model, the supplementary information, and then go all the way to runnable code? And this can be things like PopPK models, which can help with your model-based meta-analysis, but we've also been working on larger mechanistic models as well. So here, the idea is you drop in the PDF, and the first step is of a reconstruction of the model in this kind of component form. So all the way on the left, you can see the parameters are separated out from the equations and things like that. And if you hover over them, you can see where in the publication they were pulled from. So you can see, okay, this parameter came from table 2, this parameter came from, line 42 of the supplement. And anything that may warrant further review by the scientist. So, for example, if a parameter value is missing, it's included in an equation, but it's nowhere to be found in any of the parameter tables, that would get a red exclamation warning. And so the scientist needs to step in there. They can either go search the literature, try to find a reasonable value, or let the LLM make an assumption. Um, but again, that one leaves room for some errors. So it's better if the scientist kind of guides that. But those are all displayed and in a way that is not overwhelming, so it can be minimized and expanded if necessary. And then the results are validated against published figures. So the agent will go ahead, convert that model view, which is we use SBML as our intermediate, and then it will convert it into the language of choice. Um, R, MATLAB, and Python are best supported right now. And so it will go ahead and run a simulation to match the figures in the publication, and then results are plotted against each other. And then it will work iteratively. So if there's a big difference between the two, it will kind of suggest some perhaps changes to the model and allow the scientists to approve those changes or not, and then kind of work through that until the results are as aligned as possible with what was in the published figures. And then one feature kind of related to this is that all the changes to the model and associated like simulation files are automatically logged. And this is nice. One, it allows for collaboration and transparency and what happened, you can revert back. But also, this allows for some scientist oversight of the AI since it will likely make a lot of changes in trying to align the results to what was published. Here, you can have some transparency in what was changed, and this allows the scientists to review in more detail and make sure that all of the changes are easily accessible to the scientists without being overwhelming. You don't have to read the entire code file. You can just see these summarized changes. And so this is one case study. We have several others, but this is our copilot successfully replicated a pop PK of dupilumab. The agent took about 6 minutes to kind of replicate this. So you can see here we have an embedded VS Code in our software as well, so that the model file can be viewed here. And then on the right, you see our blue simulation figure with the digitized data in the red dots over top. So a pretty good fit. And then, yeah, the code can be exported and you can kind of use that for your own model building. And as I mentioned, we have done this with very large models. So QSP models with hundreds of parameters and as the model gets larger, the results it's a little bit harder to get a perfect match. We're always forgetting parameter values in our publications, unfortunately, but that just happens. But yeah, so I think that's pretty much the end of my talk. I just want to end with, we would love to work with anyone that's interested in collaboration. As I mentioned, sometimes we help companies build databases from scratch, and so we use our software and we kind of handle all the QC. But we're also opening up our software for piloting. So if that's of interest, please feel free to send me an email. And yeah, I'd love to take any questions that you may have.
Thank you, Emily. Fascinating talk. And we do have a few questions here. First one is, does the scientist in the loop method, in the initial data set identification step, automatically classify which impact a publication may have? In other words, translational, statistical, pharmacovigilance, ADME, in vitro, preclinical, et cetera, so that the appropriate subject matter experts, like the live scientists, can review.
Oh, that's a great suggestion. Currently, no. We do have a tagging system for like the type of model. So if it's a QSP model that gets tagged differently versus a pharmacometrics, and then we have some tagging for like disease areas and things like that. But that is an interesting further step. Yeah.
Thank you. So if you, let me see, how can I ask this? can the data be changed in the uh, analysis? So either by mistake or fudging data. So can you actually extract data from a paper and then change a point inadvertently or nefariously?
That's a good question. Um, Theoretically, yes, within the software, you can, once it pulls the data, you will see, like, for example, the graphs, like you'll see the points over top. The scientist does have the ability to go in and adjust those points. So in that case, you can change the data and then it would feed into the data set. Um, but hopefully that's done with good intentions, but still
Yeah, I
visually.
agree. I'm just,
Yeah.
and is there a way to audit that so that you know if something was changed from sort of the automated extraction of the point?
Yeah, that's a good point. Yeah, we do log all the changes, so it is kind of inherently auditable and we work in projects, so you can see like which user made certain changes at what time and you can revert back to previous versions of the data set as well. So yeah, those changes are logged.
Thank you. Do you have a log file for the database preparation as well? And have you tested its reproducibility? I mean, if you repeat the same prompt, will you always get the same database?
Yeah, so we do logging, and with Gen AI, there is always, they are stochastic models, so there is always a bit of, room for things to be changed, slightly or variance between the runs. So,
Hmm.
yes, but we do logging, and then, part of the process in certain steps, not all steps, but we do multi-runs. So we kind of take advantage
Hmm.
of that stochasticity to improve accuracy. So it's kind of built in as well. Yeah.
Okay, thank you. Another question. Often these databases will include both internal and external data. So, perhaps proprietary, confidential data. Um, how do you handle data privacy?
Yeah, so for internal data, what we usually do is we offer like a self-hosting option where all of this is within the firewall and kind of uses the appropriate or the agreed upon large language models, et cetera.
Hmm.
And that helps us maintain yeah, that data privacy.
So you're all, I guess, related to that question, are you also pulling in external, like public literature and things like that, to assist in the to feed the model, in addition to the confidential data which lives within the firewall?
Yeah. The external data would be pulled in and would help with model building. And we have a lot of things going on in the background as well to make sure that like modeling best practices are met as well. So yeah.
Okay. Next question. Could you talk about the service model? How is the runtime, compute power, and cost of the simulations set up in the service?
Yeah, so it definitely depends. For the kind of fee for service business model, we kind of charge per data set and we kind of handle all of the compute and things like that. And then if, the team is interested in using the software themselves, it kind of depends on what they're interested in. We take a hybrid like seat usage based model just in case someone is, a power user and they're um, on it, 8 hours a day. There's a bit more a usage charge there, but pretty flexible, and it depends
Mhm.
on what the interest is.
Is that something which you figure out up front, or is it what if you get an unanticipated heavy burden of computation for a particular set of prompts?
Yeah, so we're definitely still in the pilot stage process where we kind of use the first few months to kind of get an idea of what usage looks like and then kind
Hmm.
of use that to handle the
To
setup of
scale it, yeah, scale
yeah.
it pricing.
Yeah.
Um, this is a logistical question. How do you deal with publication access fees. So,
Yeah.
a lot of the literature is protected by copyright things that you have to buy licenses. How does that work in your models?
Yeah. So we kind of have that burden be placed on the user, if you will. So we can present the abstract, use the abstract, but then if they want the full text, they have to go through their institution to see if they have licensing. And there's a step further there where you may have access to the paper, but you may not have a specific kind of license that allows you to use GenAI over it. There's a lot of legal gray area
Mm,
there. But
mhm.
there's certain APIs that we can hit with your institution to see if you have the right kind of access before you pull it into the database builder. So,
Oh, interesting.
yeah.
Okay.
Yeah.
Okay. Um, how do you deal with a situation where, and I've experienced this, where the model code and the parameters in the published paper don't actually match the published figures in that paper. And usually, I believe this happens because there are versions that have been used to generate the figures, which get updated in the model code and parameters that are then published. How do you handle that?
Yeah, that's a very common scenario. So in that case, the LLM will present a number of suggestions. So it will say, maybe we should adjust parameter X. We could also try parameter Y. Um, and then the scientist kind of decides which one to kind of try first. And it
Mhm.
happens iteratively, and we use that model versioning to kind of see which set of changes will get us closer to that publication figure. But at we're always trying, the guardrail there is we're always trying to align first to what was in the publication. We're trying to be as faithful as
Yeah.
possible and then see which one is most likely to be a mistake, right? So there's
Mhm.
oftentimes like, simple mistakes can happen. A decimal point is off in a parameter value or something like that. So we try those first and then go from there.
Okay. What are your thoughts on benchmarking and evaluations for the applications of these tools?
Yeah, I have a lot of thoughts on benchmarking. I think for things like data set building, benchmarking is quite easy to do, right? You just do it manually, and we've done a lot of benchmarking that way as well. We've built databases by hand, and then we see, how aligned is the AI. But
Mhm.
for things like model building, that's much it's a much bigger ask to build a benchmarking set. I do think in general, as a field, we probably should be thinking about building a benchmarking set that we come together and collectively build. And we can borrow ideas from the broader AI um, landscape there, right? There's like software engineering benchmark sets that are accepted by the entire field. So
Mhm.
I think there's an opportunity, a big collaborative opportunity there for building a really rigorous benchmarking set that we can use to evaluate all of these Gen AI tools.
Thank you. Next question, similar to an earlier one. For sensitive internal document questions, can we use our own foundation model endpoints? For example, using our own internally approved Azure Open API key? And can we update this over time as newer models are released?
Yes, absolutely. So yeah, that goes back to the way we worked with some companies is self-hosting, and then yeah, we can swap out the models with the approved versions, and yeah.
Great. Okay, that concludes our questions. I'd like to thank our speaker, Emily Nieves, for an excellent presentation. Her contact information is on this slide if you have additional follow-up questions. And the recording of this webinar will be posted on the ROSA webinar website within a few days. So thank you to the audience for your participation. And please look to the ROSA website for upcoming webinars. Our next one will be in March. Thanks, everybody, and we will see you next time.
Thank you. Bye.